This next section is focused mostly on epigenetics, so talking about modifications that we can make to the nuclear architecture to expose certain areas, making them more likely to be expressed, or to condense areas, making them more likely to be silenced. So, looking specifically at heterochromatin and euchromatin, what Barr bodies are, how we can methylate and acetylate histones, not to be confused with the methylation of cytosines and what a CpG island is, and how all this plays a role in imprinting diseases. And then randomly, we're going to end it up with X-linked and trinucleotide repeat diseases, because I just didn't know exactly where else to throw all that. Your DNA can be bundled up and condensed into this large chromosome. Notice that it's kind of asymmetric, right? Above the centromere, it's a smaller half, and then below it's a larger half. So the smaller half is the P arm, and that's P for petite, and then Q is literally just the next letter in the alphabet. So the larger section is Q. So whenever we say chromatin is typically describing DNA plus the accessory proteins, typically the histones that are surrounding it. And a bit of DNA will have 8 histone proteins around them. So, we'll have 2 A's, 2 B's, 2 3's, and then 2 4's. And so that's why this is an octamer, and an octamer is really just a nucleosome. And there is technically a 9th histone protein that links between nucleosomes. So again, 8 histone proteins with DNA wrapped around it is a nucleosome. We can have the chromatin sort of spread out. That's going to be euchromatin. Then we can have the chromatin condensed, and that is going to be heterochromatin. The heterochromatin is inactive, and the euchromatin is active. And the reason for that is, if you're trying to imagine fitting a transcription factor, it's got to bind to the DNA with the DNA, it's just got to bind to the DNA. It's a lot easier in the area where the DNA is exposed in the euchromatin. It'd be a lot harder for that transcription factor to fit in here. So the histone, we call them tails. So it's like the N terminus over here, it doesn't wrap as tightly necessarily. There's going to be a lot of lysines and arginines, and that should make sense. If the DNA is negative, we'd expect a lot of positive amino acids to be on histones. But modifications can be added to these tails. So like this lysine here could be methylated. And so if you're putting something on, it's a writer. You're taking it off, it's an eraser. And if you're reading a modification and doing XYZ function, then you're a reader protein. So this is just an example of a histone 3, the 9th lysine. It has 3 methyls, and then an eraser could take something off. Now it's 2 methyls, and then of course we could go back as well. So that would just be a methyl transferase and a demethyl transferase. And so pretty soon, though, we have 8 different histone proteins, all with different tails and lots of variations, lots of possible combinations of acetyl groups and phosphates and sumoyl groups and ubiquitins and methyl groups, and it's quite enormous and mind-boggling and complex code. We don't really have to know all of that. We can just simplify it to distill these 2 basic principles. If there's a methyl group, we're going to consider that group to be off, more likely to be in the heterochromatic state. And if there's an acetyl group, it is more likely to be on, spread out in the euchromatic state. And we can actually kind of make sense of the lysine. So if you were to imagine that the histone protein has a tail that, like we said, is a lot of positive things, it's more complicated than this, but we can imagine that it's kind of wrapping a little bit on the DNA because this is a positive, and so it's going to be very attracted. But if we add an acetyl group, because of resonance, this carbonyl carbon is going to be particularly positive, and so we wouldn't want positive there. So we've abrogated the charge. And if we get rid of the charge, now we can imagine that the arms are a little bit up. Now the DNA is a little bit more spread out, and the transcription factor could now bind. So the acetylation, we could actually make sense on a like very close up chemical level. But yeah, as I said, it's a really complex code, right? And the same way that AUG is a methionine, UAG is a stop codon, there is a complex epigenetic code. In addition to the readers that are reading all of this. And of course, we don't have to know this. I don't even know if scientists have fully worked this out yet. We probably need some complex machine learning thing to really crack the code. So girls have 2 XXs, XX, and gentlemen have 1 X. But you can't, I mean, if you think of like trisomy 21 Down syndrome, an extra 50% of the 21st chromosome can have really, catastrophic effects. So, yeah, I know guys and girls are different, but you can't have two human sexes have such a dramatic difference in terms of having a whole extra chromosome. It's just not compatible. So there's got to be some way that we're making it so that people who are XX and people who have XY have a similar amount of gene transcription from that X chromosome. And so a couple possibilities is we could have more transcription in the male X, we could have less transcription in the female X, or, and this is what actually happens in our case, you can inactivate one of these X's. And these are called bar bodies. So here's a zygote, here's two functional X's, and then pretty early on during embryology as, we're forming a blastula and a gastrula and whatnot, some of the one X chromosome or the other will inactivate. And it's actually quite random, and it's not always the same one. So we end up with this complex pattern, this sort of mosaic of, X from mom compressed versus X from dad compressed. And to compress them, there's just a hyper heterochromatic state. So it's a lot of methyl groups and not a lot of acetyl groups. You could use bar bodies in this mosaic model to explain the tortoiseshell color on cat. So if we look at this cat, and we see this tan fur and this black fur, we can explain this using bar bodies. So just imagine that the purple X that we see here is for tan fur, and this other X here that's teal, it's going to be associated down the road with cells that produce the protein for black fur. And again, we're going to have these just completely random inactivations, and so this one's got its descendants will express the black color, and this one's descendants will express the tan color. And so if we kind of play that out and continue playing that out, we're going to get little pockets where some areas are black and some areas are tan. So we methylate the histone protein, but we can also methylate the cytosine. And it actually works out a little bit easy, though, because in both cases, it's going to lead to condensation, repression, silencing. So this would be methylated DNA. It's typically going to be associated with compression. A CPG island. So this is the term. So term CPG, C is just cytosine, and then G is just guanine. So what's this P here? Well, nucleotides are linked via phosphate between the 5' carbon of one sugar and the 3' carbon of the nucleotide sugar above it. So the P is just for phosphate. It's kind of stupid. We could have just called them a CG, and I think it would have made just as much sense. But that's what I mean. So it's an area in the promoter where you might have a lot of methylation. And if you have a lot of methylation, this area is going to be turned off and condensed. Imprinting. So babies are little parasites. Why? Because they're sucking all the nutrients from mom. And problematically, sometimes they suck a little bit more nutrients than is healthy from mom, right? So let's think about incentive. So if you imagine, a couple trying to sell a house and their realtor, it seems like that their incentives are aligned, right? If you sell a $300,000 house, it's a 6% fee, and half that goes to the buyer, half that goes to the agent. So at the end of the day, a $300,000 house, the agent gets $4,500, right? So the agent is incentivized, you would think, to try to make as good a sale as possible. If instead of selling the house for $300,000, we sell it for $600,000, well, then the agent makes $9,000. So obviously, again, the agent and the people selling both want to sell it for as much as possible. But if you think about it a little bit more, maybe they're not completely aligned. Because, 300,000 is 600,000. That's easy if it's just, if it's just one deal or the other. But imagine just to get an extra $10,000 for the house. It's more flyers that we got to pull up. It's longer. We have to leave it on the market. It's more calls that the realtor has to yield. It's 30% more work. It's like an extra month or two of her time to get the house for $310,000. Well, the couple definitely wants that extra $9,400 after the 6% fee goes to the realtors. But what's actually getting in the pocket of the realtor who's selling it? Just $150. After the realtor's agent, 50% is going to her broker fee. So in case the math didn't add up for you. So 30% more, she's only making an extra $150. It's just not a good use of her time. And so she actually might want to sell the house for cheaper than they do. So why am I telling this silly story? Because it kind of explains imprinting and the mom and dad's genes. They both want the baby to be as healthy as possible, right? They both want it to be healthy and fit. They want the mom to be healthy and fit because she's going to help her other child, and they both want the baby to be healthy and fit. So it seems just like the realtor and the people selling the house, there's an alignment of incentives. But the dad's genes actually, is a little bit, a fair amount less investment of resources. His ability to generate another child is fairly easy. So it might just be this tiny little crumb more in favor of stealing nutrients from mom at her detriment to the baby's benefit. And so we actually have, this misalignment of incentive. This is really the entire idea behind imprinting. So look at these two genes. These are the two real genes, the insulin growth factor 2 and H19. So insulin growth factor's job is to allow more sugar into the cell, which allows the baby to grow more. And then H19 is going to inhibit growth. And so what we see now is that for this top picture here above this line, this is somebody, a male or a female, and they are choosing to methylate, in other words, imprint this H19 gene, and they're letting the IGF-2 be normal. And then it's vice versa for the one below this line. So if you had a guess that the top chromosome is a male or a female, it would make sense that it's a male and the bottom one's a female. Because in this case, we're going to, this is on, we're going to steal more nutrients, and this is off, the inhibition of growing. And so that's exactly what we get. So if we look at another gene, P57, so P57 is going to be a CDK inhibitor. CDK is pro-growth. And so males are going to want to inhibit the inhibitor of the CDK, which is for growth. And the females are going to want to have it active because they don't want the baby to grow so much. This is the male right here. A male has one chromosome from dad. And again, the males typically want to methylate this area. So it is the paternal chromosome is methylated. The maternal chromosome, it makes sense, does not want to methylate it. She wants that open. Now, when this man generates sperm, whether the sperm comes from this chromosome or whether it comes from this chromosome during meiosis, it doesn't matter. We're going to erase all methylation programs and reset them to the male preference. So all of the male sperm, regardless of which chromosome, now have a methylation of this particular gene. And females would be the exact opposite. So again, females, though, they have one copy from dad and one copy from mom, and the dad copy, just like in the gentleman up here, is going to be methylated, and the female is not. But again, whether it's this chromosome going through or this chromosome going through, we're going to erase whatever the setting is and reset it. And the woman would like to have this gene expressed. She doesn't want the baby to grow so much inside of her and steal a lot of her nutrients. So an idea which synergizes with this relatively well is uniparental disomy. That's probably a new term to you, but you guys probably learned in undergrad the concept of non-disjunction. So during meiosis, we are going to separate the homologous chromosomes first, and then we are going to separate the sister chromatids. And if we look over here, this actually should say 2, and this should say 1. So if we look over here, everything split fine here, but we have a non-disjunction of it at meiosis 2. So we have too many chromosomes here and not enough chromosomes here. This is something that would lead to like Down syndrome, trisomy 21. And in this case over here, non-disjunction of it in meiosis 1, so that we have too many chromosomes over here and not enough over here. So that's non-disjunction. You guys know that, but what is uniparental disomy? Imagine that this, let's just say this is an egg. This egg goes up here. Excuse me. Let's say this is chromosome 17. So, the fusion happens, we get a zygote. There are now 3 copies of chromosome 17. Sometimes this will be bad and you'll get something like Down syndrome, or maybe it'll just be a spontaneous abortion. But sometimes somehow the cell can decide that it should destroy one of these chromosomes. And if it destroys this chromosome over here, we get this, which is really just maternal disomy, which means both of chromosome 17 are from mom, right? Normally everyone's chromosome 17, one from mom, one from dad. Here we have 2 from mom. Okay, well, what if we have this other situation go over here where there's just no chromosome 17? Now I only have 1 chromosome 17, and that's about as bad as having too many copies of chromosome 17. Well, again, the cell can sometimes recognize something's a little bit off, and it can duplicate this chromosome. So now we'd have a paternal disomy. So uniparental disomy is when you have a non-disjunction event, and then there's a kind of rescue, but you don't have heterogeneity to the chromosomes. This can, you could also have mutations and deletions, but I would think that this is going to be a major cause for something like imprinting. And if this occurs, imprinting typically is we're going to have mom version is off and dad version is on, or dad version is off and mom version is on. But if I have this uniparental disomy, I might have too much, both versions on, or both versions off. So let's look at the P57. Dad's is off, mom's is on, and it might turn out that just one dose of P57 is like ideal or standard, at least as the baby is an adult. If in this case we had a uniparental disomy from the male, we now have two copies of the sort of male imprinting pattern, the silencing. So we're silencing this inhibitor of growth, so we're going to have too much growth. We look at IGF-2, you could kind of have the same thing over here. So I guess this would be another male disomy. We have two, this is pro-growth. So here we have inhibition of two genes that are inhibiting growth, and here we have activation of two genes that are pro-growth. This is just too much growth. So both of these would lead to the same phenotypes of macrosomia, where the baby is too large because it's sucking too many nutrients from mom. And it's not just sucking nutrients from mom. The cells are all about taking nutrients from their surroundings and growing and dividing and growing and dividing. What does that sound like? That's cancer. And so Wilms tumor is a common renal cancer that kids get due to imprinting issues, sometimes due to mutations, deletions, often due to uniparental disomy. So Prader-Willi and Engelmann is a similar thing as well. These are more imprinting diseases. So turns out that on one long stretch of chromosome 15, we have a region, the actual genes don't matter. There's a couple genes, let's call them W, X, Y, and Z. So these are on in the male. So we would probably assume that these are somehow pro-growth, but let's just drop that point. They are turned off in the mom. They are silenced, they are imprinted, they are methylated. And then a little bit down on this chromosome, we have these two more genes, UB3 and A. UB3 you actually do have to know. So UB3 is the only one that's real you have to know. These, conversely, are silenced, methylated, repressed in the males, and expressed in the mom. So if we go ahead and look at the wild type over here, on the maternal chromosome, we have W, X, Y, and Z imprinted, which means turned off. The only genes you have that mom's bringing to the table are UB3 and A. And on the male, all the males bringing to the table is W, X, Y, and Z. A and UB3 are imprinted. That's normal. So what's happening is we're getting one functional copy, one copy of UB3 and A from mom and one copy of W, X, Y, and Z from dad. If we look at the Prader-Willi syndrome over here, so the mom chromosome is fine. UB3 and A are there, and just that's normal. But on the paternal chromosome, there's been a deletion. And if there's been a deletion, then we do not get the W, X, Y, and Z. Because remember, it's typically dad's job to bring W, X, Y, Z to the table, but we've had a deletion there. So this deletion is responsible for Prader-Willi. And I always thought of like P for paternal, Prader paternal. If you look at Engelmann, it's the opposite. We have its deletion on mom, and so the paternal copy is fine. So we don't have UB3 or A, so the maternal deletion is now responsible for this. So again, W, X, Y, and Z and A, or you don't have to know. The only thing I would know is that UB3 is on the mom, and paternal deletion is associated with Prader-Willi, and maternal deletion is associated with Engelmann. So what are the phenotypes? Prader-Willi, hyperphagia and obesity, intellectual disability, hypogonadism, and hypotonia. So there's not much muscle development. For Engelmann, seizures, ataxia, intellectual disability, and this very particular type of laugh is described as a happy puppet, which is kind of a weird way to describe it. Moving on to X-linked symptoms, ornithine transcarbamylase, Fabry, glucose 6-phosphatase, Hunter. We've already talked a bit about these, so we're going to focus on these over here. Turner's, Klinefelter's, Rett, muscular dystrophies. Note that the last one is not X-linked. I just included it because I thought it was a good idea to bundle all the muscular dystrophies together. And then Fragile X, which is a trinucleotide repeat. So Turner and Klinefelter. So this has to do with non-disjunction of the sex chromosomes. Klinefelter is XXY, and then Turner syndrome is X not. So there's not a second X chromosome, it's just one X. These are for the Klinefelters, and these are for Turner syndrome. For Turner syndrome, I would say the web neck and the shield chest are going to be real big standouts in a vignette. Now, this is a lot of characteristics. I think if you just think about, just forget about bar bodies for a second. Just assume, and it kind of makes sense, that the X chromosome might have some genes that have to do with female secondary sexual characteristics. And so for Klinefelter, if we have extra for the X, then we're gonna. So imagine that X has the height gene and sexual characteristic for females. So very tall makes sense. Not a lot of beard growth if you have less testosterone and more estrogen. Breast development because you have estrogen, X chromosome. Female pubic hair pattern, again, you're gonna have estrogen, not as much testosterone, more estrogen, testicular atrophy. And the short stature, again, we're just pretending that the X chromosome has an important gene for height, which maybe it does. If you don't have you're not going to have as much estrogen, your ovaries won't develop, and you'll have amenorrhea, and you also need your ovaries to produce estrogen, and so you have underdeveloped breast. Estrogen promotes and contributes to breast growth, and then you have these little moles randomly on the body. That one's, I don't really know. Rett syndrome, so X dominant. However, 95% of cases of Rett syndrome are just de novo mutations during gestation. And the protein here that's mutated is MECP2, and this stands for methyl CPG binding protein. And so we said that CPG islands are just, CG in the promoter. They're often hypermethylated and they get turned off. They get condensed. Well, this is actually the protein that binds. It spots all of these methyls in the promoter. It binds them. It's actually what that does the condensing. The condensing. Now, in Rett syndrome, this is dysfunctional, so you don't actually get silencing. And it turns out if boys get this, it's just going to kill them. You need to bind the CPG islands and condense them. Now, in these females, this is just a very important thing to do. So in females, they have a backup, but I guess it's not enough to fully rescue the phenotype. But boys, it's just, if you don't have any condensing of the CPG islands, then you just won't. It's an embryonic lethal. Severe motor and cognitive dysfunction will develop. Muscular dystrophy. So dystrophy just means that the muscles are going to waste away. So we have three different kinds: Duchenne's, Becker's, and myotonic muscular dystrophy. Becker and Duchenne, notice they're going to be more similar. They're both X-linked, and the myotonic is actually quite different, autosomal. So for Duchenne's, we have this dystrophin protein, which is just important for muscular stability and development. This dystrophin protein is an incredibly large gene. It's one of the largest genes in the human body. And that's why there is just a large likelihood of having a mutation here. Often they're frame shift mutations, a deletion specifically, and this might result in a truncated non-functional protein, which will contrast with Becker in a second. So it's such a large gene, you're going to have a mutation somewhere, and if it induces a frame shift, you get a shitty protein that doesn't work. Gower's sign is this very specific, because they have muscle wasting issues and not a lot of strength, to if they're on the floor, to stand up, they will stand up in this very characteristic way that they might describe in the vignette. And again, very severe phenotype, so the muscle weakness will be very young. Becker is almost the exact same thing, dystrophin protein, frame shift, but it's a functional protein. It can still do its job, just not as well. So they say that Becker is better, is a way to think of it. And it's early adulthood instead of, right when you're born, because again, the phenotype is less severe, and that makes sense because the protein's working a little bit. Myotonic dystrophy is for the DMPK gene, dystrophia myotonin protein kinase. It causes myotonia, where you can't actually, if you contract your muscles, it's hard to relax them. So the example that's often given is that if you do a handshake, it's just hard to let go, basically. People develop cataracts. There's hormonal issues. So testicular atrophy and premature balding. And this is a trinucleotide repeat. It's one of our 4 trinucleotide repeat diseases. The second trinucleotide repeat disease is Fragile X syndrome. So this is the most common the most common inherited cause of intellectual disabilities. Down syndrome is actually more common, but it is sporadic, so it doesn't count as an inherited disease. And FMR1 is the gene that we're talking about, and it stands for fragile mental retardation 1 gene, which kind of makes sense. Trinucleotide repeat, it's a CGG, and if we make another link to CPG, that those islands, they often, for whatever reason, get spotted to be methylated. So this is not a promoter. This is not a CPG island, but because we have so many CG, some system, some protein in the cell sees this and might think it's a CPG island, and it hypermethylates it, which can turn this gene off. Large testes, that actually comes up in vignettes, is a thing. Large jaws, and particularly large ears, which we can see here. This kid is just absolutely adorable. Trinucleotide repeat diseases. Huntington's is going to be really common, highly tested. We've done Fragile X and myotonic, so just Huntington's and Friedreich. For all of these, the more repeats you have, the more severe the phenotype is. So this is for Huntington's and CAG repeats. And so it's not just the more repeats you have, it also happens to be the case that typically as you accumulate repeats, it's easier for your body to produce more repeats. So over the generations, the grandpa might have 40 CAG repeats and have mild Huntington's, and then their kid will have 52 repeats and have moderate Huntington's, and their kid will have 74 CAG repeats and have a severe phenotype. That would also explain that if your phenotype is more severe, for the grandfather, the Huntington's hit them in adulthood. For the parent, a little bit earlier. And for the kid with so many CAG repeats, not only is it very severe, but it hits them very early. These terms are called anticipation. Friedreich ataxia is the GAA repeat of the frataxin gene, which is important for iron regulation in the mitochondria. It's going to present quite early. You're going to have kyphoscoliosis. So this is where you have both sagittal and coronal displacement of the spine. Ataxia, hence the name, so balance and coordination issues. Diabetes, hypertrophic cardiomyopathy, so it might be like a young person, the heart basically gets too big and you get a heart attack. And so people and athletes in their early 20s might just die, basically. And I guess if you have Friedreich ataxia, we know the phenotype, but that might just describe somebody who has a heart attack, and that's hypertrophic cardiomyopathy, and it's associated with this. Pes cavus is this super arched foot, and the hammer toes, not this big toe, but these other toes are the hammer toes. Huntington's chorea, sometimes it's called, it causes these involuntary movements, and that's because the basal ganglia, the basal ganglia has two different pathways, a direct one, which is like to initiate movement, and indirect one to inhibit movement. The indirect pathway is destroyed in the basal ganglia. So all of the movements that your body typically wants to do, but you don't even know about because your basal ganglia says, no, let's not do that movement. It'd be weird. It's now not working here. So they get this chorea phenotype presentation. It also affects their mental state, their personality, depression, aggression, and behavioral changes, and eventually leads to dementia, and hits people particularly early as well, in 30 or 35.